So welcome to our seventh ISU seminar in lymphatics. And this presentation is going to start with Riya.
All right, so hello everyone. So let's talk about the organization of the lymphatic system. So there's three basic things in it. There's the lymphatic vessels, which include like the capillaries and a few medium to small sized arteries and veins, and also like large arteries and veins too. And there is something called as a lymphoreticular tissue, which consists of the lymph nodes and the spleen, and the lymphoepithelial tissue, which consists of the thymus, appendix, tonsils, Peyer's patches. So I'll be talking about the lymph node, but before we get into the main structure of the lymph node and everything, I want to just go over a few definitions which are very important, and they are like very basic. So you need to know these things. So the first thing is that parenchyma. What is a parenchyma? It's basically this entire part of the tissue which forms like the morphological and the specific part of the organ, and it basically is a functional part of the organ. So for example, when we talk about the lymph node, the functional part, like the main part which makes up the entire lymph node, is called as the lymphatic tissue. So the lymphatic tissue is the parenchyma of the lymph node. I hope that is making sense, and I hope that is clear. The second definition which you need to know is what stroma is. So stroma is basically just the supporting tissue of the entire organ, which is made up of dense connective tissue or loose connective tissue. So just to go over both of these definitions once more, the parenchyma makes up the main functional part of the organ, whereas the stroma is just the supporting structures. So when we talk about the lymph node, the first thing you have to know is what lymph is. So lymph is basically like this colorless fluid which comes when the blood plasma kind of leaks into the interstitial spaces, and the fluid which collects there is called as lymph, and then it flows through the entire lymphatic system. The first thing it flows into is the lymph node. So the function of the lymph node is basically to filter all the lymph, and the lymph also carries like these bunch of antigens which are in our bloodstream, and so the lymph node is like the first lymph node has the first contact with all of these antigens which are present. So it also has like sort of like an immune system function. So the lymph, it enters the lymph node from the afferent lymphatic vessel, and there's a capsule on the outer side of the lymph node. So this afferent lymphatic vessel is going to kind of pierce into the capsule, and that's how it enters into the lymph node. Then it flows into like these bunch of sinuses. So there are three types of sinuses. There's a subcapsular sinus, paratrabecular sinus, and a medullary sinus. The function of these sinuses is basically they recognize the antigens which are present in the lymph, if there are any antigens present in the lymph, and they do so by because they have these cells, specific cells, which are called as lymphocytes, macrophages, or plasma cells. So these three cells are present in the sinuses, which can basically detect if the lymph has any antigens present in it. So the first sinus is the subcapsular sinus, and as the name suggests, it is the sinus located just under the capsule before the main lymphatic tissue originates. And the paratrabecular sinuses are the sinuses which are present in this part of the cortex. I hope you can see where I'm pointing. Um the last sinus is going to be the medullary sinus, which is located inside here, over here in the medulla. One more important thing which you have to know is that these sinuses, they are incomplete, and they are lined with these specific type of cells which are called as littoral cells. So the flow of the lymph is going to be pretty slow, and that is because like I said, like this part of the sinuses of the lymph node have got macrophages and everything, which are basically responsible for initiating the immune response. So the flow has to be really slow so that all of these cells can clearly like check if there is any antigen present in the lymph. And if there is any antigen present in the lymph, it is taken up by the macrophages, and then it is presented to a bunch of higher organs and everything, which decide what they want to do with this antigen and what kind of immune response has to be initiated and everything else. So let's talk about the structure of the lymph node in more detail. Like I said, it is a parenchymatous organ, so it has parenchyma and a stroma. The parenchyma of the lymph node consists of three things. There is the outer cortex, the inner cortex, and the medulla. The outer cortex is this part over here, and it has these specific circular things which you see here. They are called as the lymph nodules. So wherever there's lymph nodules present, that is going to be your outer cortex. Now, all of this other part, this does not have any lymph nodules, and it is called as the inner cortex. This pale structure over here, this is going to be your medulla. The key features about the outer cortex and the inner cortex is that the outer cortex is B dependent areas, which means that the outer cortex mainly only has B lymphocytes, whereas the inner cortex mainly has T lymphocytes. So that is a major difference between the outer cortex and the inner cortex. Now, when we talk about the stroma, like I said earlier, there is a dense connective tissue capsule on like the surface of the lymph node, and there is also something called as a trabeculae, which are made up of the dense connective tissue. They basically come inside the lymph node, and they kind of break the lymph node into like separate structures. So when we talk about the outer cortex, like I said earlier, it is a part of the lymph node which consists of these lymphatic nodules. There's two types of lymphatic nodules. There's a primary and the secondary. So the primary lymphatic nodules, they mostly contain B lymphocytes, but they also have T lymphocytes and macrophages. And when this primary lymphatic nodule, there are antigens present, the lymphocytes kind of get activated, and then it forms this light colored area which is called as a germinal center inside the primary lymphatic nodule. And when this germinal center is formed, that's when the lymphatic nodule is called a secondary lymphatic nodule. So the germinal center basically contains these large cells which are called as the lymphoblasts, and they arise when the lymphocytes undergo this special process which is called as blastoid transformation. This is very important to know because this can very easily be asked in your micros, and for that matter even in a slide tests, which you when they will ask you probably to explain secondary lymphatic nodules and how it arises. So this blastoid transformation occurs, which basically converts the lymphocytes into lymphoblasts. And this transformation basically kind of helps produce more memory cells and more plasma cells. And if there is any antigen present, so like a proper immune response can take place. But if there are no antigens present, the germinal center kind of disappears, and the lymph nodule again becomes into a primary lymphatic nodule. But the germinal center can keep on reoccurring again and again depending upon if there is an antigen present or not. So this light structure over here is a germinal center. Just to recap quickly, it contains lymphoblasts, and this region outside, which is kind of forming a cap to the germinal center, is called as the mental zone. This mental zone consists of B lymphocytes, and it sometimes also has memory cells. There are these other type of specific cells which are called as the follicular dendritic cells, and the only important thing about them which you have to know is that they have these irregular processes which form a 3D network. And since it forms a 3D network, it can basically trap all the antigens which are present. And since it can trap antigens, it's going to have lymphocytes present in it too. So think about it this way, that whichever organ is coming in contact with the antigens, they're always going to have lymphocytes and macrophages and all of these things, because these cells are responsible for initiating the immune response. The paracortex is like the inner part of the cortex, which does not have the lymphatic nodules. And it is, like I said earlier, it is a T lymphocyte dependent area. And it also has these other accessory cells, such as interdigitating cells. The paracortex, it also has these specific blood vessels, which are called as high endothelial venules. And these endothelial venules, basically, they have got these tall cuboidal cells, and they are a bit sticky on the surface because they kind of allow the lymphocytes to enter into the lymph node, to the bloodstream. This is the main way the lymphocytes can get into the lymph node. The lymphocytes, when they move into the paracortex, they move through the spaces between the cells, which is called as diapedesis, or they enter directly through the cells, which is called as the transcellular route. The T cells, they always stay in the paracortex, whereas the B lymphocytes, they move into the outer cortex. Like I said earlier, because the germinal center and everything, they are mainly B lymphocyte dependent areas. The the reason why these high endothelial venules have tall cuboidal cells is because it helps loss of fluid during this whole process. The medulla, it consists of two structures. So there are the medullary cords, which are these dark, irregular bands, and they mostly contain plasma cells. They are also the site of maturation of the plasma cells, and they produce the antibodies, which then, along with the lymph, they go into the bloodstream. And these white structures here, they are the medullary sinuses through which the lymph flows. And multiple of these medullary sinuses, they join together to form an efferent lymphatic vessel. So many people usually get confused between afferent and efferent. So think about it this way. Efferent lymphatic vessels, it starts with an E, so it is going to exit the lymph nodule. And I hope this was clear, and I'll let Varun start with his topic. Does anyone have any questions?
All right, if no one has questions, we'll begin with spleen. So let's start with basic functions of spleen. It is involved in filtering of the blood. It traps foreign antigens from the blood. It helps the immune system respond to threats in the blood, and it acts as like the storage of blood, so blood reservoir, and it produces blood cells during hematopoiesis. So when we talk about the structure of the spleen, it's divided into two basic parts. We have stroma, which forms the framework of it, and parenchyma, which forms the functional part of it. So when we talk about the stroma, it is made up of capsule, trabeculae, as well as reticular connective tissue. So when we talk about the capsule, it is made up of dense connective tissue, which you see on this outside. It's made up of dense connective tissue as well as smooth muscle cells. And then we have the trabeculae, which are the strands of connective tissue which are protruding inside the lumen. So it will protrude inside. So they will come from the capsule and they will protrude inside the spleen. And then we have the reticular connective tissue framework, which holds everything together. When we talk about the parenchyma, it's divided into two parts. We have the red pulp and the white pulp. So the red pulp is for the blood storage as well as filtration, and the white pulp plays a main role in immune function. So just like a basic idea about it is that spleen is like a big sponge that cleans and stores the blood. And it has just two main parts inside. One is for cleaning and one is for storage. Yeah, so moving on, when we talk about red pulp, it has two components. We have Billroth's cords and then we have splenic sinusoids. When we talk about Billroth's cords, it's nothing but 3D network of reticular connective tissue made up of reticular cells and fibers. So we had talked about the reticular cells in the first two seminars about the structure and features, and if you want to revise that, you can. And when we talk about the functions of the Billroth's cords, they fill the spaces between the neighboring sinusoids in the spleen. Now, when we talk about splenic sinusoids, they are just like irregularly shaped capillaries which have spindle-like endothelial cells. And the main function of these nuclei of these endothelial cells is that it protrudes into the lumen inside the inner cavity. And they're externally supported by the circular process of reticular cells, and there are gaps between the endothelial cells that make these splenic sinusoids, which help them have a porous appearance, which means that their wall will have some gaps. So this will allow the blood cells to move through. And once the blood basically moves through the venous sinusoids, then it empties into the pulpar veins, same to the venules. When we talk about the splenic macrophages, so as I talked about the structure of the sinusoids, these are made up of endothelial cells with a porous wall, and they allow the blood cells to pass through. And then these openings are flexible, which are just based on the pressure. When we talk about the role of these macrophages, I think everyone knows about this. They're involved in phagocytosis of old or damaged cells, and they contain hemosiderin, which is a brown pigment which is obtained when hemoglobin is broken down. And the origin of these macrophages is from they're derived from monocytes, and the function is same: engulf and digest foreign materials and act as antigen-presenting cells. When we talk about the splenic blood circulation, so yeah, this is the entire chain of it. You guys can make some mnemonic to memorize this, like it's pretty long. So it starts from splenic artery, then it goes to the trabecular artery, then we have the central artery, then penicillary arterioles, which have multiple branches, then sheathed arterioles, which are surrounded by lymphatic tissue, then we have arterial capillaries, then sinusoids, and as I said before, from sinusoid, the blood drains into the pulpar vein, then trabecular vein, and splenic vein. So out of these, all of these blood vessels, the four main ones are the central arterioles, penicillary arterioles, sheathed arterioles, and atrial capillaries. So when we talk about the central arteries, they move from the trabeculae. So they move from this part, this trabeculae, into the lymphatic tissue, which is the white pulp, and they eventually then branch into the penicillary arterioles. When we talk about the penicillary arterioles, they just represent a transition point between white and red pulp. And then when we talk about the sheathed arterioles, they're enclosed by the lymphatic tissue, which is involved in filtering of the blood as well. And then when we talk about the arterial capillaries, they empty directly into the reticular meshwork of the red pulp. So now moving on to the last part of the spleen, we have the white pulp, and it has the periarterial lymphatic sheath, lymphatic nodules, as well as marginal zone. So what does all of these terms mean? So first, when we talk about the periarterial lymphatic sheath, it is this part which is around the central artery. So you can just imagine this. I'll explain with the slide in the after this, okay? So this is the central artery, and then we have the periarterial lymphatic system around this, and this contains the T lymphocytes. So this is why we call it the T-dependent area. And then we have the lymphoid nodules, which are represented by the light areas here. So these are the largest accumulations of the lymphatic tissue, and they contain mainly B lymphocytes, okay? And when we talk about the marginal zone, it acts just as, so this part, it acts just as a boundary between the red pulp, which is outside, and this part is the white pulp, which is inside. And the function is, it is key area for immune response. Lymphocytes, they leave the sinusoids and they enter the white pulp. And it contains antigen presenting cells, B lymphocytes, plasma cells, as well as medium sized lymphocytes. So guys, here I have attached like a YouTube video which helped me understand spleen. When I first studied spleen, it was really annoying to study because there was so much going on. And now I'll explain you guys with the slide here, just to like recap everything. So first, as I mentioned, the spleen, it is surrounded by a fibrous capsule, which is made up of dense connective tissue. So here you can see, you can find this on your fingerland. This is labeled by the department itself. As you can see, this is the dense connective tissue capsule. And now just the basic way to identify the slide is, you can see these light areas. These are the lymphoid nodules, secondary lymphoid nodules, and all this red area is the red pulp. So the red pulp would be just the, as I said, Billrod's cords and venous sinusoids, and all of this represents that. Now, when we talk about the white pulp, as I mentioned, these are the different structures. So first of all, as I said, this is this periarterial lymphatic sheath, and this part represents the central artery. So central artery in the center, surrounded by the periarterial lymphatic sheath. And remember, this is the T-dependent area. And then moving on to the next of it, we have the lymphatic follicular here. And then this is surrounded by the marginal zone, which again acts as a transition between the red pulp and the white pulp. So this is about it, and all of these are just, and yeah, when I talked about trabeculae, these are the trabeculae, and you can see these are just the strands of the outer capsule which protrude inside the spleen, and you can see they have like, a spindle type arrangement of the nuclei of the cells. So that was about spleen.
Okay, does anyone have any questions for what we just did? If not, it's okay. We're going to move on. So now we're going to talk about the thymus. Sorry that my voice is a little bit, I lost a bit of my voice. So just let me know if you guys can't hear me or need me to repeat something. So this is the thymus. There's an image on the top right corner. You guys can see where it's located. And the thymus is the primary lymphoid organ or the central lymphoid organ. And it's very important that you guys know this because you will be asked, like, what is the primary lymphoid organ? And you guys will answer it's the thymus. And I will explain to you guys why the thymus is primary and why the other organs that we talked about are secondary. I just want to let you guys know the thymus is a very important slide. I got it for like my credit tests when we used to do slide tests for them. I got it for my final, and multiple times professors will just ask you questions about the thymus whenever they feel like, they just want to see if you know it or not during a final. For some strange reason, I don't know why, they really, really like the thymus. And I will tell you guys like later, show you guys the structures to be able to identify the thymus, which is very important. So try your best to pay attention. So the thymus is important. It's known as the primary lymphoid organ because the T lymphocytes, which you guys know or will know, which are the cells that like help fight infection and protect our body. The T lymphocytes, they're created in the bone marrow, but they mature into immunocompetent cells or like into the mature cells in the thymus. So once they go to the thymus, they mature and then they're ready to like protect us and fight all these antigens. And this is known as immunological surveillance, or the thymus is responsible for immunological surveillance. Because of the thymus, we have a proper immune system. And so how this starts, it starts with precursor cells that are produced in the bone marrow. And from the bone marrow, these cells are going to go into the cortex. And I will show you guys how the cortex looks like in a second, but just try to visualize. They're going to the thymic cortex. And when they go into the cortex, they're going to settle in the extracellular space. And there, the lymphocytes are going to start to proliferate and mature. So again, the lymphocytes are going to come from the bone marrow. They're going to go to the thymic cortex. Then they're going to start proliferating, and then they're going to become mature. These lymphocytes will contain receptors, and these receptors are going to differ in their specificity. Now, these receptors are generated spontaneously without there being an exogenous antigen. The receptors are very important because with the receptors, the cells are going to be able to like recognize antigens. The antigens will bind to the receptors. So in the thymus, so again, one of the things that happen in the thymus, the lymphocytes come, they proliferate, they mature. Second, they're going to develop their receptors, which is important for them to recognize the antigen. And through that, like you have this large repertoire of lymphocytes that's produced. Thirdly, we have cells. The cells that are produced like this, they can exhibit reactivity that's directed against the self-antigens, meaning that they can attack, like if they're not created properly, they can actually start attacking our own cells. Now, there's a whole other process that I will explain in a second that will that's going to basically like prevent that from happening. But this is just theory wise. Theory wise, we have receptors, and because these receptors are so different, we're going to create many different lymphocytes. And these lymphocytes, however, are going to be very reactive. Okay, they have the ability to attack our own self antigens instead of just attacking the exogenous antigens. Before they can leave the thymus, in order for them not to attack our own antigens, they're going to undergo what's called the immunological surveillance of the thymus. So just try to remember like these things that happen in the thymus. They proliferate and mature. Because of the receptors, we have a lot of different lymphocytes. However, these lymphocytes can attack our own antigens, so they need to go through immunological surveillance, which is the selection process. So the immunological surveillance or selection process, basically, long story short, guys, the cells that are attacking our own self-antigens or that have the ability to attack our own self-antigens or our own cells, they're just going to be destroyed. While the cells that we know that are not going to do that, we are then going to send them to the secondary organs, like the lymph nodes, the tonsils, etc. So we have the positive selection. So the positive selection means that we see that we look at cells that have receptors that can bind to self-MHC molecules. This means that these cells, they can tolerate our own self-antigens. They will look at our MHC molecules and they will not attack it. Because remember, guys, lymphocytes are there to destroy, like you have lymphocytes that are going to destroy cells, right? So it's important that these lymphocytes don't destroy our own cells. So the lymphocytes that have receptors, because remember, right, they have many different receptors, like there's a large receptor specificity. The lymphocytes that the receptors bind to our self-MHC molecules, they tolerate it, they don't destroy it. These are the cells that we want, right? Because that means that they're not going to attack us, but they're going to attack every like exogenous thing. So these cells, they receive a positive signal, and the lymphocytes that do not fulfill this requirement, they're going to be eliminated by apoptosis. You have the negative selection, and the negative selection does not permit further development of the autoreactive thymocytes. And any T lymphocytes that's carrying a TCR, which is the receptor, that can bind to the self molecules, like our own exogenous antigens, is going to be eliminated. And basically, guys, just know that out of all the lymphocytes, right, that are being like mature, 95% of those are autoreactive, meaning they're going to, like, they don't pass the selection. They're essentially destroyed. Only 5% of the T lymphocytes will actually fulfill the selection criteria, and then they're going to migrate. Remember, they went to the thymic cortex. They proliferated, they matured, they got their receptors, they went through the negative and positive selection, so we destroyed all of the autoreactive ones, and all of this is happening in the cortex. After they leave the cortex, they migrate to the corticomedullary zone. And from the corticomedullary zone, they're going to cross the venules and enter the bloodstream and go to the secondary lymphoid organs. And in the secondary lymphoid organs, this is very important. Where do they stay in the secondary lymphoid organs? They stay in the thymus-dependent areas. So for the lymph nodes, this is the paracortical zone. For the spleen, this is the periarterial lymphatic sheaths and also Peyer's patches. Do you guys understand like why these cells that were were created in the bone marrow, matured in the thymus? Do you guys know why they're traveling to like other places? Why don't they just stay in the thymus? Can anyone guess?
No, don't know.
Because lymphocytes, right, guys, this is our immune system. It wants to protect us, right? And when we come in contact with antigens or like bacteria and stuff like that, we want to make sure that our lymphocytes are essentially are going to be able to come in contact with that. So that being like through I don't know, through our GIT, through blood or whatever, we want to make sure that we have lymphocytes spread out to kind of be able to attack these antigens. So we're not going to keep our lymphocytes in just one place, which is the thymus. We want to make sure that they're in the spleen. We want to make sure that they're in the lymph nodes. We want to make sure that they're in Peyer's patches, which you guys don't know what it is yet, but you guys are going to learn what they are in GIT. We just want to make sure that, you know, our immune system, in order for it to be perfect, it can't be in one place only. So that's why there's kind of like this transition. Just, it's really important that you guys know they're made in the bone marrow, they mature in the thymus. They mature in the thymus, and in the thymus, they will go through this selective procedure. And it's important that you guys mention the selective procedure, because the selective procedure, when there's problems in the selective procedure, guys, this is what can lead to like autoimmune diseases or other diseases, because you have lymphocytes attacking, you have your immune system attacking your own cells instead of just attacking the bacteria from the outside and exogenous antigens. So the selection procedure is important, and you guys need to talk about it. So now we're going to actually just like histo-wise look at the actual thymus. So here's a picture, but I will show you guys in the histology fingerland thing. The cortex is this part. It's the dark part. And in the cortex, you have a lot of small T lymphocytes. While in the medulla, which is the central pale part, you have less lymphocytes, but they're larger. Can anyone tell me why? It has to do with like what is happening in the cortex versus what is happening in the medulla. So we said that in the cortex, right, we have the immature lymphocytes, right? They come from the bone marrow, they settle in the cortex where they proliferate and they mature. So we have a dense accumulation of small T lymphocytes in the cortex. They're going to ask you guys, why are they small? They're small because they haven't matured yet. They haven't proliferated, or they are proliferating. That's why it's dense, but they're small because they haven't matured. And why do we have less lymphocytes in the medulla? Well, because we said 95% of the lymphocytes don't pass the selection procedure. So only a small amount of the lymphocytes are actually going to go to the cortical medullary area. So they then travel through the blood vessel to the other secondary lymphoid organs. So the thymus, guys, if I remember correctly, it was actually, there was another structure, I don't remember what it was, another slide, that it was easy for us to kind of confuse it with the thymus. The one thing that you guys are going to see this thing and you 100% know that you guys are in the thymus, you're looking at the thymus, is this structure known as hustus corpuscles. The hustus corpuscle is just a concentrically arranged flattened cell reticular epithelium structure. It's connected by desmosomes, and the central cells undergo hyaline degeneration. And the hustus corpuscles, they help eliminate the self-reactive lymphocytes. I'm going to show you guys what they look like, but just know that when you see it, you are 100% in the thymus. Okay. So this is how one of the slides for the thymus looks like for you guys. And so I'm just going to zoom in, and you guys can see we have very dark areas, which you guys, we just spoke about, are the cortex. And then you have the very light areas that does not have as much accumulation, and you'll see that the cells are much bigger. Compare the size of this cells to like these, much smaller. And so cortex, medulla. And hustus corpuscles are these concentrically arranged, hold on, apologies for this, structure. Let me see. I believe this is one, but usually they're much better visible. So essentially, yeah, you're looking for one, and and some slides, the professors will always let you know that some slides, the way they're prepared, you're not going to be able to see certain structures, but for your actual slide test, they will not give you those slides. Guys, so these are the hustus corpuscles. So they're very easy to like differentiate from the actual like cortex and medulla. You're not going to look at this and think this is a lymphocyte. So this is a hustus corpuscle. This is a hustus corpuscle. Here it is again. I believe this one too. Here's another one. Here's another one. Here's another one. You know, they're essentially everywhere. And this is the thymus. It's very simple. You just are going to see the hustus corpuscles. You're going to see dark and light area, and you know you're looking at the thymus. Now, you guys are actually going to, so this is again the thymus. Now you guys are actually going to be and you can see here better the cortex and the medulla. So again, you guys are actually going to have two different types of thymus. This thymus that I just talked to you about or spoke to you about is known as the juvenile thymus. Basically the thymus that's found in kids. When you guys undergo puberty, something happens to your thymus. And this is called or termed involution of thymus. So the thymus is fully developed at birth, but when you guys get to puberty, it's going to reach its largest size, and then it's going to undergo what we call involution. Basically, the parenchyma is going to be replaced with adipose cells. And instead of you having the normal like medulla and cortex, you're just going to have these small islands of functional parenchyma that you can see. This next part, this little like paragraph here, is extremely important because they will ask you guys, what are the main characteristics of the like involution of thymus? What do you see? And you guys need to be able to say these things. First, it loses definition as a distinct organ. It's going to decrease in weight. The lobulation will disappear. The boundary between the cortex and the medulla is going to disappear. The septa will become thick. The size of the hustus corpuscles will increase, and the production of T lymphocytes will decrease. You guys need to be able to say that, like, we have the normal thymus, and then we have the involution of thymus, or involuted thymus. And in the involuted thymus, it looks different. Basically, the parenchyma disappears. You have a lot of adipose cells. You can't differentiate between cortex and medulla. It decreases in weight. There's no more lobules. And again, septa thickens. However, note that the involuted thymus is still considered a functional lymphatic organ. This is how the involuted thymus looks like. As you guys see, there's a lot more white. We have a lot more adipose tissue going around. Here, and we have some islands of parenchyma. Like this would be an island, this would be an island, this would be an island, compared to what we were seeing before, which was this entire thing was with the parenchyma. Now, theoretically, it does say that the size and number of the hustus corpuscles increase, but this also really depends on the slide and how they prepare it. I know that I received slides where that was not the case, but theoretically, know that the number and size of the hustus corpuscles increases. Guys, if you guys recognize the thymus, there's no way that you guys are going to be confused between a juvenile thymus and an involuted thymus. I mean, look at this. You guys cannot differentiate between the cortex and medulla here. You just can't. And there's so many adipose cells everywhere. If you look at a juvenile thymus, it is much more well structured. You see the different lobules. You can easily differentiate between medulla and cortex. There isn't like adipose tissue, as much of the adipose tissue. And essentially, you would have less hustus corpuscles, but again, that really depends. So very clear differentiation. And if you guys like see it right now, then you guys are good to go for your credits or slide tests with the thymus, pertaining the thymus. So yes, this is how it looks like. And then just some extra information. We're almost done. Know that the thymus is a lymphoepithelial organ. It means that it's composed of lymphocytes, lympho, and reticular epithelium. So lymphoepithelial organ. And you guys haven't done embryology yet, but know that the thymus originates from two sources. They might ask you this, and you will get extra brownie points if you know this. I can't say for sure if they will fail you if you don't know this because you haven't done embryo, but they will be very happy if you're able to answer this. If they ask you, where does the thymus, what two sources does the thymus originate from? And the lymphocytes, right? Because it's a lymphoepithelial organ. It's lymphocytes and the reticular epithelium. The lymphocytes are going to come from mesenchyma. The epithelium part is going to develop from endoderm of third pharyngeal pouch. I know that doesn't make any sense right now, but it will when you guys do embryo. Okay, note that now let's talk a bit about the microenvironment of the thymic cortex. Remember, guys, the cortex is where the immature cells first go, proliferate, mature, gain their receptors, and undergo the selection procedure. Important to know these things. So the microenvironment, we know that there isn't an exogenous antigen, right? And there isn't exogenous antigens because we don't have afferent lymphatics going there. So I'm not sure if you guys have actually studied the immune system PPT that you guys are supposed to have, but yeah. So there's absence of afferent lymphatics, and there is the blood thymus barrier, which I will talk about in a second. So the thymocytes, they have contact with our self-antigens, right? They need to have contact with our self-antigens because we need to see if they're going to like become reactive or not. Because if they're reactive, we need to destroy them. So they need to come in contact with our self-antigens, and they come in contact with our self-antigens on the surface epithelial cytoreticulum. And in the microenvironment of the thymic cortex, you also have some hormone production. However, wouldn't say this is as important. Blood-thymus barrier. Basically, the blood-thymus barrier, it shields the T lymphocytes from circulating gas. Here's just a picture. This is what it's composed of. I'm not going to read it out loud. Like, you guys are just going to have to memorize it, and the picture is going to help visualize it. And that was all for thymus. And last thing that we're going to talk about are tonsils, just very, very small and quick. And tonsils are these like small structures, and they contain an arrangement of lymphatic tissue. They're usually found in the entrances of the digestive and respiratory system. You guys are going to look at palatine tonsil, lingual tonsil, and sublingual tonsils. If I'm not sure because they change it every year or year to year. But the palatine and lingual tonsils, they form mucosas. The mucosa will form invaginations, and you're going to have these crypts, tubular crypts. And this is going to cause like food and bacteria to kind of like get stuck there, if that makes sense. And then the lymphocytes that are in the tonsils that came from like the thymus, they go to the tonsils and all these other secondary lymphoid organs. We spoke about it. They will then like attack that bacteria. So you guys are gonna study the palatine tonsil, and this is how it looks like. A lot of times when you guys get given the palatine tonsil, you guys get given this exact slide. Like, this is exactly how it looks like. So you just need to memorize how it looks like. And the palatine tonsils are found on either side of the pharynx. They're covered with stratified squamous non-keratinized epithelium. And you have these crypts, the crypts that are lined with the same type of epithelium, but the crypts are infiltrated with lymphocytes, so that debris and bacteria will accumulate in the crypts and the lymphocytes will attack it. And note that the palatine tonsil has a capsule and dense connective tissue making up the septa. Then you have the lingual tonsil and nasopharyngeal tonsil. Lingual tonsils are in the posterior third of the tongue. They're also stratified squamous non-keratinized, also have crypts that food and bacteria get stuck in, dense connective tissue, forming septa. There's absolutely no capsule, and you will see glands in the lingual tonsil. You will see glands. For us, the nasopharyngeal tonsil, we needed to learn the theory. However, I don't think there was a slide for nasopharyngeal tonsil. There is, but I don't remember us doing it. Anyways, so this is the lingual tonsil. As you guys can see, this is the tonsils and the glands. I mean, the entire thing is the tonsils, but really what you can differentiate it is these glands. And then here we have the pharyngeal. I'm still not sure if you guys might receive this, but check your book because I'm sure we did not receive this. And then lastly, that's it. Yeah. Palatine, lingual, and nasopharyngeal. Okay, guys. Any questions? No? Okay. It was good having you guys. Wait, yeah?
No, nothing.
Okay. So see you guys.
Thank
you
you.
for coming.
Thank you for explaining. Toodles.